Welcome to Quantum Photonics. We are here with an article which we had discussed in the newsroom, and also this particular study elicited a lot of interest in some of my posts in social media and created, in fact, many discussions among different sectors. So, this new study published in Molecular Psychiatry had provided groundbreaking insights into the biological underpinnings of autism, revealing that adults with autism have fewer synapses in their brains than neurotypical individuals. Using a type of brain imaging called positron emission tomography or PET scan, researchers measured synaptic density directly on living individuals for the first time. They discovered a strong link between reduced synaptic density and difficulties in social interaction and communication, offering a potential explanation for some of the challenges faced by autistic people. Autism spectrum disorder is a complex condition that affects communication, behavior, and social interaction. Although it is well established that genetic and environmental factors play roles in autism, the biological mechanisms behind its features remain unclear. Many researchers have speculated that synapses, the connections that allow brain cells to communicate, are altered in autism. Previous studies have suggested this possibility based on animal models and post-mortem brain tissue, but these methods provide only indirect evidence. To address this gap, the research team at Yale School of Medicine aimed to directly measure synaptic density on living individuals with autism. They hope to recover whether reduced synapses might be a factor in the social and communicative challenges experienced by autistic people, as well as to explore how these changes might inform future therapeutic approaches. So, I would like to introduce our guest speakers here. Um, Dr. David Matuskey, who is a board certified psychiatrist and neuropsychiatrist with research work that has been translational in nature and focused on elucidating the underlying pathology of brain conditions such as autism spectrum disorder or ASD, substance abuse, and neuropsychiatric disorders like, for example, Parkinson's disease, with an aim to find effective clinical treatments guided by molecular neuroimaging. Studies have included using in vivo PET imaging to investigate the role of neuroreceptors such as dopamine D3, serotonin 1B and G, and McClure-5 and kappa opioid systems, neuroinflammation, and most recently, synaptic density, SV2A. In clinical and non-clinical populations, ongoing interests include imaging, neuropsychiatric and addictive disorders, and the demographic, social, and environmental factors influencing our brain. Then our guest, Dr. Adam Naples, is PhD, is an assistant professor in the Child Study Center at the Yale School of Medicine. As a researcher at Yale, he has co-authored papers on autism, reading disability, and genetics, and developed novel experimental methods for studying brain activity during live and simulated social interactions. Dr. Naples received his BS from Cornell University, his PhD in psychology from Yale University, and his postdoctoral training at the Yale Child Study Center. He has also been active in mentoring and training of graduate and undergraduate students and the postdoctoral fellows. His primary research interests are understanding the neural and cognitive mechanisms that lead to variability in developmental disorders. I'd like to present to you Dr. Adam Nichols and Dr. David Matuskey. Welcome to Quantum Photonics, Adam and David.
Thank you for that fantastic introduction, Cecile.
Yes, absolutely. I think you've done our work for us. Thanks a lot.
Yes, so we are very excited and someone because here in Quantum Photonics we usually have a newsroom once a week to update people who are interested about what's happening in science and tech. And yeah, I think I shared this on November 19, your article in Quantum Photonics, and it has received a lot of interest from the audience, and as well as Kevin, one of the regular supporters of Quantum Photonics, said, "Did you discuss this in the newsroom?" Then I said, "Yes," meaning this upcoming room that we have with you. Then he said, "I thought I'm going crazy." And anyway, because we are excited, we'd like to give the floor to you, David and Adam.
Yeah, thank you so much for having us on. We're happy to talk about the study and any of the details that are there. I don't know, honestly, that the way it's kind of set up. I know that there's PowerPoint slides there. Is that something that people just read by themselves, or is that something that I should go over like a slide deck?
Yeah, you can go over it like a slide deck. You try
Okay.
to communicate or sync with people by informing them about the slide numbers. So, for example, you will say you're in slide 9, and this is what it says. And or
Okay.
you can say we're in slide 9, but please also take reference to slide 2, because there is a relationship of what I said there to this slide, so people will just surf their way. And if I presume it's going to be presented in a chronological manner, you can just mention the slide numbers on a case to case, from time to time, not for every slide.
Okay, that sounds good. So, Adam, maybe I can start since this is more in the pet realm initially, and then I'll
Yeah,
kick the ball off to you as soon as we get to autism and let you kind of take us all the way home here.
cool.
Um,
I'm just pulling them up. Take it
all right,
away.
sounds good. All right, so I'll pull it up on my laptop here. And so I start with really slide 2 here, and so I gave this I give a talk like this basically every year to high school students and other folks who are learning neuroscience. And I admit I'm a bit of a philosopher. I'm certainly a traveler, and so if you look at some of those slides, they're really interesting places like Sri Lanka. There's Croatia there. There's a slide of Kyrgyzstan where I was in a yurt, and it turned out that the lake that's behind there, you can see it just a little bit, turned out to be the origin of the Black Plague in the medieval ages. I did not know that at the time. That came out in Nature a couple years after I left, but that I actually had that as my screensaver because it was the only time in my adult life, I felt like I had nothing to do. I was just riding horses and running around, and it was kind of a great thing. But when we look at all these experiences, I think there's a commonality, right, for all of us, and that is really what gives us those experiences, and that's our brain. And so sometimes it's called a 3 pound universe, and there it is. But how do we understand it? How do we get into that? And I think that's where it's been a very long, arduous road in science to try to really understand what's going on in there. And spoiler alert, like we still don't know, right? We're still trying to understand exactly what's there. But here on slide 4, I think I try to take folks just through a really, really quick kind of tour just to get some folks up to speed. And so it turns out we're Adam and I are over here in New Haven, Connecticut. And I just saw that the Peabody Museum today, they emailed, has a new exhibit on some of the origins of neuroscience, which is really cool. And so Golgi and Ramon Cahill initially were two important neuroscientists at the time, and they had kind of competing models about how do neurons work. And so you can see that like on the left there, there's like a black image there. And so it turns out that Golgi was wrong. He kind of thought that that neurons all bunched together in these large networks, and that they weren't individual. And it turns out that they are individuals. So that's kind of like the origin, that sheet of the idea of really how a neuron looks, but also somehow how it behaved. And you can see the cartoon there. And so basically, neurons are just brain cells, right? And they have a part of the cell that connects to another neuron. It's called a synapse. And those little gaps you can see in an electron microscopy kind of blown up with some interesting looking dyes. So people can really take a look at that and see, wow, our thoughts, our emotions, yes, our experiences are really done through those kinds of connections, right? So that's kind of the base of those things. We don't have the ability in modern imaging and modern medicine to look at things at that level. These are done in the animal models. They are done when folks die, and they're able to like look at stains and understand those things. And so that's a bit of a black box, though. We can't get there. And so what can we do? So I have an image there of medical imaging, and so there's different ways to image the brain in living creatures and living people. And so one of those I'm sure everyone has heard of, MRI, right? So if someone has a fall or might need to get checked out, there's a question of a concussion or something, they'll often will get a CT, computerized tomography, which is basically like an X-ray of the brain, or they'll get an MRI, which is magnetic resonance imaging, and they're both quite good at what they do in a lot of ways. They give really good resolution, and you can look at the underlining structural, parts of the brain and understand is there a severe or an acute injury, right? And there on the right side, there's one with little red dots on it, and it's called a functional magnetic imaging, where you can look at things like blood flow essentially and see whether or not the blood flow shifts in different parts of the brain. And the idea is that, well, if that part of the brain is being used more, there's more glucose, there's more blood, there's more oxygen, and therefore that's become more active in whatever task or activity that the brain is involved in there. And then there's other kinds of interesting, most of them are MRI modalities where you have something like diffusion tensor imaging, where you're able to look at the white matter tracts, or in some ways, the roads, right? The highway of the brain, when all those axons line up and kind of allow a certain direction to happen. So those are all great modalities and they have a lot of strengths, but they don't tell us some of the underpinnings molecularly of what's going on. And that's where PET imaging, or positron emission tomography kind of comes into play. I'm going to skip through slide 6. It's supposed to rotate, but it probably won't do that for you now. And then 7, which is another similar one. PET can be used not just for the brain, for other parts of the body too, but we're just going to really focus on the brain. And so in slide 8, it has a very, very simple diagram of kind of what this technology is. But essentially what it is that there's a small amount of a medicine per se. In this case, we call it a tracer, or sometimes it's called a ligand, so a radio tracer or radio ligand. And what happens is that there's a group of chemists. Sometimes they like to be called alchemists because that's really what they're doing. They're really changing the structure of this little bits of this medicine, and they're able to give it a little bit of a tag. Most often it's carbon 11 or it's fluorine 18. And what happens is it gives a signal. And so really what happens is, because those are isotopes, is that there's a bit of energy that's given off. It's a predictable, it's a known amount of that energy, and it comes in the form of a positron. So that's where the positron emission tomography comes from. And what happens with the positron is antimatter. They're actually making antimatter. And then antimatter and matter don't really like each other too much. And so then when they meet, they annihilate each other. And that's where the energy is given off. And then through a collection of sensors, and my favorite word in all of PET imaging, and that's coincidence detectors, you're able to find out where exactly in the brain that happened to a fairly good resolution, not the resolution of a couple slides before. You can look at neurons. We're talking maybe, at the very best, we're lucky enough at Yale to have that. It's going to be about 1 to 2 millimeters of resolution. In this study, it was about 3. So that's kind of like the level of science we can get to spatially. And so then we're able to like say, okay, fine. Now we can get this tracer in the body, we can get it into the brain, we can start to maybe think about the molecular underpinnings of the brain. And I just have on slides 9 and 10 some examples of some of the math or the modeling that occurs for us to be able to get those answers, right? And so from that, I like to call it sometimes the menu we have over here at Yale, but it, as opposed to something like an MRI where you really kind of have standard sequences, and PET imaging, every time you inject that little bit of tracer into somebody, it really is its own science. It's its own math. All those things are done, and you're able to look at very specific um, molecular underpinnings, and they might have absolutely nothing to do with the scan that happened before. For instance, you could look at something like dopamine, and the next scan you might look at serotonin, and the next one you might look at glucose, and the next one you might look at something like um, you know, um, synapses. How about that? And so, why would you want to look at synapses, right? And on slide 12, I show that in some postmortem work. And so you can see in something like Alzheimer's disease or epilepsy or things like depression and things like autism, right? And schizophrenia, that clearly synapses are affected. As a neuropsychiatrist, someone who tries to devote my working life into the brain, I can't think of a condition or ailment in the brain that doesn't involve synapses. They all do. And so I think that's what got, that's what can be really exciting about a synaptic marker. This allows people really to study so many different things and normal and the abnormal elements of the brain. So how do we do that? And so on slide 13 here, there's a cartoon there, and so we go back into the synapse, which we mentioned before. And so it turns out that when these synapses, sometimes when I think about it, it's almost like trick or treating or a little bit out of the season now. But there's a bunch of like goodies that are packed together, right? And it doesn't matter what kind of neuron it is. It could be glutamate or dopamine or serotonin. It doesn't really matter. And so they all get bundled up together in like that, kind of like that Halloween bucket. And then someone like knocks on the door, and basically the bucket goes out the window, and all those like candies and goodies go flying with it. That's my image, at least, of what's happening in the brain. Probably it's literally happening in the brain when you eat Halloween candy. And so that vesicle, right, or that small circular sphere that packs up all those goodies, has to have some elaborate dance and some mechanism to be able to kind of package itself and go near the end of one neuron, bind to the membrane, and release all those things. And so that's what the synaptic vesicle glycoprotein 2, that's a mouthful, isn't it? So we just call it SV2A. It's really likely one of those adhesion or docking molecules that really is able to stick to that side. And because all synapses have it, it is relatively preserved from an evolutionary standpoint. It gives us an ability to be able to come up with a kind of standard way to look at synapses. And that's kind of the idea of it. In slide 14, I'm actually probably going to skip this outside of the idea of like living cartoon, which is probably a little bit more accurate of what SV2A in a vesicle looks like. We have, millions and millions of these things, probably billions of these things, actually billions, that are in the brain. That's kind of what's floating around us as I'm speaking, and hopefully you guys are listening. The slide 15 there just shows you some of the actual machine that we use. It's called a high resolution research tomograph. The molecule that was involved in this study, in this case, it was UCBJ. And then on the right side is some of the radiochemistry. Those alchemists down there were able to actually make this compound right before it's given to folks. And so I think on the next slide here on 16, you can see some of the images. Again, some mathematical modeling that occurs to be able to give us reliable estimates. But you can see we call it the BPND parametric image there, but that's kind of the resolution of what we're able to find. And then on 17 is kind of a similar one, just showing that if we give this in somebody who doesn't have an issue, we usually call them healthy controls, and we scan them one day and then scan them the next day, it's pretty consistent. And that's kind of like a bedrock of PET imaging. We have to make sure that these are reliable, and we call it test retest validity is very solid. And in this case, it was. And so if all those things happen there on page 18, you're able to get into Science Translational Medicine on the cover. We're really excited because this was the first time that we were able to publish this and show some of those images and some of the things we were able to do. And then on page or slide 19 rather, I did mention this before, just in a neuropsychiatry realm, but you can see just some of the different conditions and disorders that we've been looking at. And so that's, folks like Adam and myself, but actually there's a whole collection of people at Yale and now across the world who are using this technology to look at lots and lots of different conditions, things like Alzheimer's disease, Parkinson's, depression, essential tremor, Huntington's disease, bipolar, PTSD, schizophrenia, drug abuse, alcohol abuse, traumatic brain injury, stroke, things like that. So I'm on slide 20 now. Now I'm going to park it, have my friend and colleague Adam try to take over. It's all
Okay,
yours.
and cool. So thanks, Dave. That is just like such awesome science. So I'm just gonna briefly just go to sort of slide 21 here, talking about autism and synaptic density. So Dave, excuse me, I have a cold. So Dave, I think gave really the really important introduction. Like everything in the brain is about synapses, right? Like synapses are, well, and neurons and synapses are how the neurons communicate and connect with each other. But when we talk about autism or we talk about any other kind of conditions Dave mentioned there, we the way we know about those conditions and we learn about them and the experiences that people have, they're people and they're having their experiences and their behaviors. So we classify and we diagnose conditions like autism, behaviorally, right? So it's briefly about autism. It's a complex condition. We know there's strong genetic contributions associated with it, um, meaning if there's a person, an autistic person, has a younger sibling, that sibling is more likely also to be autistic than just like some random person kid off we might pick out of a crowd. Um, and so that's led to a lot of research in trying to understand what the genetic causes and underpinnings of autism are. And a lot of the genes that have kind of been interesting and or we've seen to have more differences in autism are associated with like synaptic strength, like the numbers of synapses, the way sort of neurons scaffold and organize themselves in the brain. Um, and so this sort of last statement here is this, even though most cases of autism are idiopathic. So what does that mean? That's kind of a lot of a mouthful. It's kind of saying is that most of the time autism's not it's not something that like a doctor can tie down to a specific gene or a specific like maybe event that might have happened in development or something like that. So we're really trying to understand, like what's happening in the brain that's associated with autism. So, all right, and then here, these are some old aims, and I can see a spelling error I made in here already, but this is kind of the idea we had going into this project is try to understand, like, look, just what does synaptic density in cortical, and that's sort of the more newly evolved part of the brain. Like when you look at a picture of a brain, the outside of the brain is usually what we're thinking about, the cortical and subcortical areas, which is like kind of the old, like sometimes people call it like the lizard brain or like the more primitive areas of the brain. How are those gonna be different in autistic people and neurotypical people? And we had some ideas about which brain regions might be the ones that would be the most different. And we had those ideas based on prior research using other methods that David mentioned, things like MRI or post-mortem studies where, after someone dies, we can look at brain and we can actually look at sort of slices of brain and look at look at the cellular level to see differences. Um, and so that's just like a big group difference. That's like we have 10 people over here and 10 people over here. And what's like kind of the difference between all the, I mean, this group on the left and this group on the right. But we also wanted to know is there an association with the kinds of differences autistic people have in behavior and sort of clinical presentation and synaptic density? Like, is more synapses or fewer synapses associated with something specifically? And so what we found was actually pretty, and I'm down on slide 23 right now, and that's a that's a figure from our paper. And I'm gonna explain it, but if people have questions, I'm happy to answer them. I think both Dave and I are. So this is it's a graphic, and it's got a lot of dots on it, and the dots are we got some, they're sort of arranged from left to right based on the brain area that we're looking at. And the y-axis, the up and down, that's the synaptic density. And each one of those little dots is a person. And on left to right is the brain area that we're measuring it in. So these are a lot of names like pallidum, the thalamus, the amygdala, the caudate. And you'll notice that some of these dots are hollow and some of them are filled in. And so when they're filled in, right, when there's a solid dots, that's a brain area where we found a difference, like a statistically significant, meaning that the size of this difference is very unlikely to have happened just by chance or by, just by random like variability among people, where we found that synaptic density was reduced or lower in autistic people compared to the neurotypical controls. And this picture is organized so that in the left, we see this thing called the pallidum, which in general doesn't actually have that many I mean, it has a lot of synapses, but not nearly as many as some other parts of the brain. And so you can see that the groups aren't meaningfully different down there, but there's also not that many synapses altogether down there. I should have mentioned that the red circles are the neurotypical group and the blue circles are the autistic individuals. And the big circles are the average of each group. But as we start going to the right, we start to see, as synaptic density goes up, we start to find that the groups are different and more consistently. And so now I'm going to go down to slide 24. And that's so this is kind of like it's like a whole brain view. It's a nice way of looking at the brain. And what we did here is we took sort of each one of those little 3 millimeter chunks of the brain that Dave mentioned before, and if there was a really big difference um, between autistic and neurotypical people here, we colored in that region of the brain to demonstrate it. And this is kind of an interesting picture. This is most of the brain shows the differences, but these are all this is the only ones that are shown in color here are the ones that are sort of like the extra big difference between the groups. Excuse me. Now going down to 25. So okay, so that's so 23 and 24 I just talked about. Those were showing that there's overall between these two groups, um, pretty meaningfully large differences in synaptic density. And then 25, I'm just going to introduce you to this tool that we use in diagnosing autism. It's called the Autism Diagnostic Observation Schedule. It's really like the gold standard diagnostic tool for autism diagnosis. And the way it works is it's a sort of semi-structured observation and it really like interview and interaction with a clinician. Like the people who like several other authors on this paper, and there's a lot of authors, a lot of people who worked on this. They're really experts on administering assessments like this and working with people like clinical psychologists, working with autistic people. What they do is they basically interview and interact with somebody in a structured kind of way for about an hour, and they assess and measure things like social interaction and eye contact, gesture, conversational turns, and this is like the number one tool. This is for doing a diagnosis of autism, but we also get kind of a measure of like some people use the word like kind of autism severity, but are just sort of like autism like kind of the you get a number out of this, right? Like, which is like just over the threshold of autism or like, further over the threshold. And so we took this number, and this is now just an autistic group, and we look to see, this is our second aim, does this associate with synaptic density? And this picture here on slide 26 is showing like kind of slices through the brain. I always think about it like, um, it's it's always reminds me of sort of like these science fiction movies from the 90s, but all the places where it's colored in are where there is a strong relationship between synaptic density and social performance or social features on the ADOS-2 in the autistic participants. And these numbers, like a correlation, the highest it can go is is 1 or -1 here because it's a negative correlation. And what this means is that, the more synaptic density was lower, the higher people were scoring on autistic features on the ADOS-2. Um, and so what's interesting about this picture is that there's color everywhere in it, in all the brain areas where there's synapses. And usually you wouldn't see color everywhere in many other kinds of studies. We would only see, we would kind of like look at one area and say, oh, look, it's over there in the amygdala or the parahippocampal gyrus or something. But what was really like amazing when we were looking at these data is that this correlation was really everywhere in the brain. And on slide 27, sort of another way of looking at that, it's like a scatter plot. And each one of these little squares is the correlation or the plot of synaptic density, and that's the y-axis going up and down, and then the score on the ADOS and the x-axis going from left to right. You can see in all these brain areas, I mean, there's more brain areas than just these ones, but these were sort of our main, where we placed our bets brain areas. To look at differences. It's a really strong correlation, and it's also really consistent across everybody in the study. So one thing that I think people always want to see when you say, hey, we saw there's higher synaptic density was associated with lower scores, they say, well, maybe there's like one or two people who have really 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 low scores or something, and it's pulling it out. But here we can see it's consistent across the whole group of people. And that's a pretty um that's for those of us who look at, like, the kinds of numbers associating changes in brain activity or differences in brain structure, with really what's behavior, right? Like the ADOS is we're working with people, and people do things, and we move around, and we talk, and we look at each other, and that's what we're measuring, right? But we do that with like a person in a psychology clinic. Um, and then we're finding, I think, this really amazing way of seeing something about how people are really moving around and making their way through the world has something really strongly associated with this measure of synaptic density that we got from PET imaging. And that's, I mean, it's a really great result. I'm going to now sort of pull Dave back in here just to also acknowledge, like, really the tremendous amount of like work this was from so many people, um especially James McPartland there, um and our funders, and really the participants, and the people who came in to participate in these studies too. It's just great. But yeah, I went to slide 28. I realized that you guys aren't looking at the slides as I'm flipping through them, but slide 28 is where we thank everyone. So yeah. So I guess now, how do we sort of open up to questions now? Or
Yeah, Adam, thank you so much. And thank you so much, David. We would like to announce to the audience that we will be opening up the floor for questions. Please raise your hand or write on the um, room chat if you have questions. And we will request our speakers to address such questions. But in the meantime, before the questions, I think David wanted to say something.
oh yeah.
No, I was just gonna thank Adam. It's a great job there going through all that data, and then just to kind of follow along Adam's coattails there. It really is a team effort. It's everyone here. I think when you do this kind of science, something like PET imaging, it's too big for one person. That so that there's chemists, there's mathematicians, engineers, doctors, nurses. Like it's just a whole group of people. I think that's what to me makes it, I always say, so frustrating and so exciting that you can get this high level of science done. I grew up in Florida, and so I tell people when I was a kid, I used to see the space shuttle launch, and we used to like actually would stop class, like elementary school, middle school, and run outside and see when they're launching that thing up. And every time we get one of these studies completed, I feel the same way. Like it's that amount of science and expertise from across disciplines that make this happen. So I'm just it feels pretty spectacular to be a part of it.
Yeah, I mean, I was thinking about that the space shuttle, like something like that. I'm in my attic right now, and we have a giant Lego rocket ship that my son and I built up here. I was looking at it, and I mean, so it's I think maybe we sort of maybe we didn't emphasize it enough, but it's in the title and in the paper. Like, so what we're looking at here is these are the synapses, right? Like this is the sort of the synapses are the connections between the neurons. It's like the highway of activity for the traffic of like thought. Um, in people who are alive and coming in and they're coming in and doing our study. This is like it's um it's pretty amazing. Like the we've thought about these things and like we know synapses are important from all the work, in these like historically that Dave showed these old drawings of synapses. But we're like, well, what's going on with like, all of us as we're sort of going about our days, like these are people and we're behaving. And so being able to measure these in people who are really alive and out and about doing stuff is phenomenal. Like we often I think like in other kinds of imaging modalities we're really we talk about things like neural computations a lot, and neurons and synapses, but this is we're actually really seeing it for the first time here in autism. It's pretty awesome.
Thanks, sir. Great. Yes. Any questions that either one of us can try to answer for anybody?
Yeah, thank you so much. I would like to ask a question. Um, do you plan to have another study? Because I understand this is done for the adult autistic people, maybe for a small sample actually, but it's fantastic because it's the first pet scan that was done for them, and also it's in vivo. So, but I was wondering, looking at what you have on page 12 of your slides, actually, of course, this is just informative, the slides that you gave us, but I was very curious. You were trying to show us pruning of autism and schizophrenia from at birth and then at 6 years old and at 14 years old. And of course, we know very well that when someone becomes an adolescent, that's when pruning comes, even for neurotypical and neuroatypical individuals. So I am just very interested, for the sake of curiosity, if you are going to do some kind of study looking at uh, PET scan in vivo. I know it's maybe
Yeah.
expensive, but yeah,
Yeah.
I'm very curious, if that's going to happen.
Yeah, right. So you're right. It is expensive and it's very intensive. And so the short answer is yes, we hope so. And so science works on ideas, but then kind of money or grants to be able to fund those ideas. And so we're trying to work through that right now to try to say, okay, can we do that to kind of go through our next step? And we have a couple ideas about where to go with that. One of them just shows that at birth, 6 years and 14 years old. Right now, this technology is not utilized much at all for children, and that's due to the amount of radiation. Even it's a very very small amount of radiation, and kids that bar is even higher, and that's probably appropriate, right? Like everyone wants to be incredibly cautious with this, and so we're looking at things to be able like new technologies we're trying to work on to develop to be able to push that bar a little bit where everyone would feel safe and comfortable putting it into younger kids. So that's one angle of it. We have other angles too that we're trying to do. In addition to that, there are other groups in the world who are working on this, particularly in Europe. And so we're hopeful that, and Canada actually also. And so we're hopeful that they publish their own data. As soon as they can to try to add to the literature. Overall, from what our understanding is that their findings are very, very similar to ours, and so that even though we have a small sample size, that's kind of like in some ways, that the known secret in PET imaging. We tend to have small sample sizes because everything's too expensive. But if you have different groups with similar kinds of results, it gives you a lot more confidence, probably correct. I did see that someone else is like Jonathan K. I'm gonna hit the invite button. I'm new to
Adam,
this, but yeah.
we
Oh,
will try to manage as to who will speak first among those who came on stage. So
okay.
don't worry about the order of the speakers who will speak.
Okay. Great.
Okay. Anyway, yeah, so yeah, I'm very excited about this, and that's why I asked that question because when optogenetics started and also CRISPR even was starting, it seems as if nothing was happening even in neuroscience for quite some time, even in the last maybe 10 to 12 years, and then something happens and it's explosive. And I thought that if you said other people in the world are trying to make a study related to the study that you have. And if we could get more data at different stages and, yeah, probably get other samples to try to encourage diversity as well. And if we could get this data, probably we could use AI also to help us, just like what happened in the Nobel Prize.
Yeah, I mean,
Yeah.
it's almost a topic I'm sure by itself, but AI is definitely sweeping the world, and science is no different there. So yes, there's ideas certainly that AI could do, with the data, to acquire it, but then also when you have the data, right? So you might be able to, in theory, come up with much more complicated models than folks like Adam and I trying to figure out equation by equation. You can do this kind of in one fell swoop. And so it's an exciting time to be in science for various reasons, but that's certainly one of them.
Yeah, thank you. I think some people would like to ask their own questions, but I just like to give a side comment. Also, aside from the usual, implications that we have about science, it I was thinking also about its implications on polygenic scoring because I have friends who have autistic children, and I was wondering that you already know that. Your first child is autistic, and then you aim for the next child. And I actually have a friend who has three who are in that spectrum. And yeah, I thought that probably, there's supposed to be some kind of counseling, although there are comments against polygenic scoring also. But still, it is something to be considered as well because you said a while ago that it could run in the family in the same way that other traits could. All right, so I would like to give the mic to Kevin. We are doing this question and answer portion in terms of PTR order. So PTR means who came up the stage first. So Kevin, who is also one of our moderators here in Quantum Photonics. Thank you. Kevin, please ask your question.
Oh yeah, yeah. Like Cecile said, yeah, she shared this article in one of our newsrooms, and yeah, it's saying, yeah. Speaking of psychology, I thought I was going crazy. I'm like, didn't you share one of these? Yeah, but my I think my main questions I'm thinking of. I missed a little bit of this, but you know, did you? I mean, do you think like glial cells, and myelin, do you think you can get more information from those and and some other parts of the brain and then were these densities in certain parts of the main like cerebrum and cerebellum and the brainstem and then I was wondering if actually as well we had a room about like cerebral cerebral spinal fluid that cushions the brain. I was wondering if you can get some markers from that, and some meninges, and so yeah, so questions like that is what seemed to come to mind.
Okay, nice questions, Kevin. Yeah, you got the whole brain in there, don't you? You got like white matter and meninges and CSF and
Yeah,
everything.
yeah.
Um, right, right. And so, I mean, short answer is those are good questions. And so, the white matter, we kind of glossed over that, but in fact, there are synapses in white matter too. It's just less of those. And so, yes, like say with this tracer, this technology, you're able to see that. If you look at glial cells, there's some synapses on those, but we can't really distinguish. We can't pull those out with this tracer per se. And so this is what's interesting about PET, right? It depends on your target. And so, are there, some targets on glia? Yes, and there's not a great PET tracer right now for that. We have some. One's called PBR28, where you look at microglia. I've published some papers before. If you're interested, you can hunt those down. But we're looking for new ways with PET to be able to look at like glia effectively. There are probably, it's unfortunately a stepchild in some ways with the brain. They should have a little bit more of the spotlight because they are terribly interesting. But I think that with this one, I would say it definitely focuses on gray matter more because you can really kind of see those differences with that. With CSF, usually what happens is and these kinds of studies, I mean, there's the practical side of science, right? And so, sure, we can go and have a doc go and do a spinal tap and be able to pull some of that CSF fluid. And in fact, I've been involved in studies that do that, but it's hard, right? A lot of folks aren't, pulling up both of their arms to say, sure, sure, I'm next to have that done. And so I think that kind of limits it in a lot of ways. But I think it's certainly interesting. There's a lot of different proteins and markers that could be in there. But again, it's just kind of like what questions you ask, what technology do you have to be able to do that. This is just one small piece, but hopefully it's a nice step forward. Just trying to understand maybe some of the pathology of autism. And just aside, I think one autism certainly not unique in this case, but I think that when you look at something like autism, it is a human condition. And so there's lots of great animal research, and I certainly don't want to besmirch any of it, but you question about the validity of some of those models exactly into the human model. And so I think that's why it's important to be able to validate that in human beings. Again, it's great, and there's things you can do in animal models that you can never do in humans. And so, in an ideal world, they really help each other. But this step, even if it's been shown in like preclinical animal models, is important. Like we have to get that evidence in humans as well, and that's where something like PET comes into play. Thank you.
Yeah, thank you. Kevin, if you have other questions, we'll go back to you in a while. In the meantime, let's give the chance to ask a question to Jonathan. And we notice you, Shane. We will, after Jonathan, we will read your questions on the room chat. Jonathan, mic to you.
Hi. Wonderful study. Really thorough and fascinating. So, my question has actually been asked by others as well, as it turns out, in the chat section, which is that in the deck that was shared, slides 23 and 27 just appear as blank white. The data is missing. If maybe at some point one of you could post the full slides as they were intended to display, that would be appreciated.
So hang on, let me just look at those ones
Was it
because
supposed to be a movie or a video?
No, I think
so
there's a data slides. Maybe there was just an error there, and when I uploaded it, possibly.
Mm.
yeah, I think I know what it is. So those I think hang on, give me one. I bet I could
Yeah,
sort
it's
of
the autism data slide and the
got it. So okay,
associations.
those two slides are actually in so if you those are the autism data is in the paper, which so if you go to the link
Ah,
in the paper, that and I think what happened there is I tried to make those, and this is now just like sort of technical fun stuff. I tried to make those nice PDF figures so they look pretty, but I think Google Slides has a problem with that.
mm.
So those are the figures in the that we have in the paper.
Okay. And I apologize if you said this, Mike.
Oh
I'm
yeah.
way up in the mountains and my connection is terrible. And perhaps you said this. Could you explain what voxels are?
Sure. They're well. I'll do my best, and then Dave was gonna probably come in and save us out here. But so you think like your brain is like this 3D thing, right? It's a physical thing. But when you're measuring activity or synapses in a certain region, where we have to kind of you know. It's almost like they're little 3D cubes that your brain. It's all like I think about it like little Legos. Like, okay, if we were going to make your brain out of Legos, little tiny Legos, because those voxels, they're kind of like the size of the Lego, that the Lego size we can measure synaptic density in.
Mm,
It's sort of like another, I think the name is actually like a 3D pixel.
mm,
Like you think about a pixel on a computer screen
hmm.
or on a screen, it's like it's a square, right? So if you zoom in really, really, really close, when you zoom out, it looks like a nice picture and it's rich, but you get in there and it's like, oh, just lots of little squares. Well, a voxel, it's a cube. And that's sort of the unit that we measure in synaptic density here, but in lots of imaging, in 3D imaging modalities. Um, you can only sort of zoom in so much, and that's as much as we can zoom in here.
Okay, so it's basically a three-dimensional Cartesian grid, and the size of the voxels is a function of the particular measuring modality.
That's, I think, actually much better than I could have ever said it, but yeah, exactly.
Okay, hey, thank you. I'll have another much more general question after everybody else's. I don't want to take time from the group. Thank you.
Yeah,
Yeah,
thank
thank
you.
you, Jonathan. We'll go back to you shortly. In the meantime, we'd like to read the question of Shane Kerpata here at the room chat. So Shane said, did you notice anything interesting in the outlier MRIs of the sample population? Uh, eh, Not MRIs, yeah. Did you notice anything interesting in the outlier scans, maybe, of the sample population? Which regions glowed the most?
Ah, so I I'll start and then I'll let Dave finish this one. Um, what I think what I noticed, and so Dave's the pet expert. I deal more in sort of other imaging modalities and cognitive neuroscience. But what what seemed most surprising was that kind of all we saw the same pattern in almost all of the brain regions. Whereas in a lot of studies in different kinds of imaging modalities, w- you, people tend to notice or identify differences in one or two or a few sort of specific brain regions, and that was what we initially thought we might see. But the results we see seem consistent throughout the brain. Dave?
Yeah, yeah, thanks, Adam. Yeah, so I guess it depends on like the definition of an outlier, right? I mean, and so that is kind of defined different ways for different folks. And so in general in science, I usually use the rule of thumb, it's beyond three standard deviations. We don't have that here. So I just as a technical point say probably we don't have outliers, but we do have a spread of data, and I think that's fair. And so I think anytime we use PET imaging, that is basically the standard. And so I mentioned a little bit before about like test retest validity, or you do the same scan twice and how close is it together? And about 10% is pretty good. And so you figure there's just noise in some of this, technology and some of this data. And so we see a little bit in there, but it's pretty close overall. And I agree with Adam completely. I think the thing that I think surprised us, right? And it surprised us, to me from a pet perspective. I know with Adam and Jamie from an autism perspective, as well as me too, was that the findings were everywhere. When you do a study like this, you're not sure. Are there going to be any differences at all? Maybe, maybe not. Where some regions going to go up and some would go down, that would be probably my hypothesis of what happened. And we didn't see that. And so I think the consistency of the data and not every region is statistically significant, but every region does go down. And so I think that really says something to there, and it goes into like maybe a basic mechanism of what's underneath, and we didn't anticipate that.
Yeah, and as Dave mentioned, like the test retest reliability is huge when you think like, is the tool we're using like a good and sort of effective tool, like a good. You think about like a ruler or a tape measure. It's got like the most test retest reliability. Like if you use a ruler, like in your attic or your basement, it's always going to give you the same measurements. But also, as Dave mentioned, sort of some other groups using the same approach or finding the same results. And that kind of like independent replication is really um is really like just you love to see it because you can always wonder, like, well, maybe there's some just something about this study or that study, or that might have uh here, maybe it was just like the 12 people who came in and did the study here in New Haven. But it seems like it's a pretty consistent finding, like consistent across the brain, and then not just in this collection of people who came in.
Yeah, thank you. So there is a question here by Anna, and yeah, she wrote, could lower synaptic density be a biomarker for autism severity? And how might this be used in clinical practice or diagnostics?
So I think this is the kind of thing when we think about like biomarkers, sort of like objective biological measures. This is kind of fits the bill, right? Like it's as Dave described it. Like you come in and we kind of put your head into this really amazing piece of technology, and we can look at the synaptic density. As far as clinical practice goes, Dave's the expert there and how PET would go used for clinical practice.
Yeah, and so I have dreams, right? And so, yeah, but part of my like dreams are just the idea that, one day you could go into an office like myself. I'm a psychiatrist and neuropsychiatrist, and that someone could have a problem, and that we could put them in a scanner and be able to understand better what's going on. We can do that a little bit already, right? We can do that with like Parkinson's disease. You can do that with Alzheimer's disease, but can it spread into something like autism? And if I parse out for a second, which I never like to do, neurology from psychiatry. Neurology is more able to do this right now as we speak, right? Like, you can do this. Psychiatry has been hard, right? It's been harder to understand what are the elements of depression, of anxiety, of autism. And so I think that the hope is that, yeah, at some point we'll be able to do that. Can we do that based on the data we have? I mean, it's too soon, right? It's only 12 subjects. It's very new. But I think that there's some potential in this. And I think one of the interesting things about synaptic density imaging that we alluded to before is the idea that it can be used in kind of every condition to some extent, right? And so, are there certain patterns in the brain that this can pick up really well? Can we put this into an AI algorithm, for instance, and be able to tell us percent probability of a certain diagnosis or disease? Yeah, I think that's in the realm of possibilities, and so it's not going to show up at your doctor's office anytime soon here. But I think that's kind of the golden path ahead, if possible, right? Is to be able to use this to be able to eventually get it into the clinic. That's like the number one goal of my research career, honestly, right? And that's a very tough bar to hit. You know what I mean? To be able to do anything where it matters, kind of in an immediate and concrete way, in a clinical office like that. That's awesome. So anyway, not now, but hopefully someday.
Yeah, thank you so much, Adam. So, yeah, Kevin and Jonathan, if you have more questions, now is the time to ask. So, let's start at Kevin first, if you are prepared.
Okay. Yeah. One thought that came is do you think this can be applied to animal models? And I think quantum computing got brought up for simulations, but that's in the future. And it's but I mean, and yeah. So comparison to animals, and if it can be applied and modeled there and looked at, and then maybe also, do you think your research can be scaled up or expanded? I know this is very early, and then collaborations or partnerships, to advance the research, and
Yeah.
yeah.
Yeah, good questions there. And so, can it be, can we collaborate? Absolutely. We love collaborating. We like to see that. I think the reality is like there's, steps in research, right? And so, often there's smaller groups and smaller studies like the ones we have. And then, there's bigger studies and bigger amounts of people or groups. And then you're able to show that. And I think those steps have to happen before it can kind of get into like the full clinic realm. The second question, or I guess the first question, really, on animal studies, absolutely. I think that's one of the powers of PET, right? Is that you can do this in rodents, you can do this in baboons, and so you can do this in a lot of different animals, and even in theory, like different kinds of there was one study I saw on a plant. I don't know exactly how that works, but and so you can scale this. I think that's what's so good, right? A lot of the animal um, you know uh, metrics or technologies they use are really awesome, but they're not going to be used in humans, right? You just can't ethically do those things. And this thing you can. So I think that's, often they call it translational science, right? There's like basic science, there's clinical science, and we're kind of in the middle. We're the bridge, hopefully, that they can kind of pull those things together.
Yeah, Jonathan?
Yeah, and Jonathan went to Yale too, guys. No pressure.
Um, so this is a very general, more neurobiology question, which is why I wanted to wait till towards the end when everyone's specific questions pertaining directly to your study had been asked. But since this group is named Quantum Photonics, I simply must ask. There's credible evidence that processes of photosynthesis and magnetic field navigation in birds are mediated by quantum level phenomena. What does current research in neurobiology indicate about quantum effectuation of ion channel function or other nerve cell activity?
All right, Jonathan from Yale, you're putting us far afield here.
Yeah.
So that I cannot answer that credibly at all. I have to admit. And so yes, I do know a little bit, maybe as much as you or less, on the idea of like bird's navigation, and the idea that clearly that there might be some quantum kind of entanglement that they're able to detect on some kind of level. I can't speak to that when you get into the humans. The one thing I would say though, because I do see a fair amount of like, quantum mind stuff, and I'm very open-minded, but as a scientist, you do have to be suspicious. And,
Mm-hmm.
and so I think that to me, I'm very big onto like to the idea of parsimony, right? Occam's edge, and just the idea of okay, we understand some about neurons and connections, and do we have to go into kind of the quantum realm to pull those things out? I have not seen credible evidence in humans that that is the case, and it might turn out to be the case in X amount of years. But right now, I don't see it. I think that quantum. I mean, I'm interested in it too, right? Like I read all these articles about it, and it's fascinating, and it clearly is getting, more important in the idea of quantum computing and those kinds of things. It's also kind of a catch-all in some ways,
Mm-hmm.
kind of like AI for whatever you want it to be. You know what I mean? There's a quantum mysticism that's there,
Mm-hmm.
and so as a scientist, I'm curious of those things, but I kind of stay away. I think our job is to kind of go step by step through all these things, and that's kind of what this study is. It's another step in that direction.
Totally understood, and I agree about the hand waviness about
Yeah,
some of the speculation.
right.
Yeah, I would
Thank you.
just like to say that several years ago, probably 3 years ago, we had a room about the navigation of birds, and of course about the question of Jonathan. We just have to look for it. It was some time ago, maybe 600 rooms ago, that we had that with the scientists who had actually done that paper. And if I don't know if I remember it correctly, Bob, maybe a year and a half ago, we had this room with the quantum biologist who cannot stop talking about her research and really went deep and is continuing to go deep on this topic. I think she came from UCLA. And then there's another quantum biologist who went into space to try to check the effect of microgravity as well, and to also test the theories um, of quantum mechanics in physiology. So
Yeah.
we will look for that. We were not able to transcribe it to YouTube because those were maybe earlier days, and I think we just have a few YouTube videos now. But we will look for it, and probably Jonathan, you can listen to this recording, and probably we can invite them again because they would answer your questions in great detail. And Bob, the scientist from UCLA, is was. Female scientist, I think. Yeah. So we will look for it and we'll send you a copy. It was like a room which lasted for about almost 3 hours.
Thank you.
Wow. Yeah,
Wow.
I'd be happy to listen to that. I'd be happy to discuss quantum brain, potentials there too. But again, I haven't personally seen anything, at least in the human brain, to say that. But I'd love to be proven wrong.
Or to be proven right. Yes, so there's another question here from Rick. How do your findings relate to the broader theory of synaptopathy in neurodevelopmental disorders beyond autism, such as schizophrenia or intellectual disabilities? And I would like to add also for this, these are the problems, but on the other hand, it's also a problem for those people who are highly gifted. I'd like to ask that because they are also neuroatypical. So, yeah. So I will repeat his question. How do these findings relate to the broader theory of synaptopathy in neurodevelopmental disorders beyond autism, such as schizophrenia or intellectual disabilities?
Do you want to go at them, or should I tackle it?
Yeah, I'll start. And as you guys can tell, Dave and I have like some overlap in our expertise and some and so we'll just sort of riff off each other here. Um, it's a a great question. I think that you know what the thing that I think of most of that question is our understanding of sort of synaptic density in across different kinds of conditions is indirect still for many of them, right? And it's I think that's why this study, as David and I said, we're like, wow, we saw these results and we were not really expecting them. Um, and so I think to what extent we kind of need to approach the different kinds of conditions with these same kinds of methods in order to make those connections. People have brought up animal models a few times here, and one thing that's I think kind of important to mention is like these are the people who came in for this study. These are adults, right? Like they were kids, they were adolescents, now that they're adults, and that's that brain of an adult who's like 25, 30 is different in a lot of ways than the brain of a of like a 10-year-old or a 5-year-old or of a 50-year-old or a 60-year-old. And we know that aging and development are very they have widespread changes throughout the brain. So when I think about like how these findings relate to other conditions, my first question is like, well, so what do we really know now about this sort of synaptics, about synapses in vivo, like in living people in these other conditions? And to what extent do we know, like, what do we know about them, depending on kind of where they are in development too? Cecile, as you mentioned, right, in adolescence is when the onset of, many things kind of can happen. Um, and so, like, really kind of getting a clear picture across conditions, I think is critical using the same approach, because we can, we know now that, we get sort of unique information from this UCBJ imaging that we weren't getting in other approaches. Uh,
Yeah. So I think this is a very landmark study. I was actually attacked by some people from the high IQ groups because I posted that, and because they don't have enough understanding about what autism really is. This is just a lot of people discussing without any academic or empirical backup, based on what they knew maybe 20 years ago, which is so different from what is known now. So, meaning, why did I post that? Because, of course, I made an introductory narrative, which is actually different from what I posted. I posted both your study but at the same time I posted a popular article because your study would be very difficult for a lot of people to grasp. And they said there was a small sample, and they said it was kind of insulting because maybe I was presuming that a lot of people in these high IQ groups were saying that they are slightly autistic because they are smart. And yeah, in my own experience, those who are actually very, very smart do not say that they are smart, but they felt slighted, and they felt not included because of them feeling that they might be in the spectrum, and because they are in that spectrum, they could be really, really outstanding or bright individuals. So yeah, I'd like to get your comments on that. Of course, I defended our ground, I think, not so badly.
Yeah, I mean, I don't think that I think that there's plenty of autistic people who are, brilliant, very cognitively able. I don't think our study is really speaks to that uh kind of question. I'm sorry that people uh, sorry it came across that way. And maybe it was in the popular press article. Um, I think this is. I mean, it's like we just got to get more data, and then they're right. Like, it's a small sample, and we can speak to that sample, but I guess that's maybe sort of just to sort of err on the side of being um is sort of simplicity is. I think that's why we need to do the studies, is to answer those other questions rather than sort of sort of extrapolate from one study to maybe a different group of people.
Yeah, just like what they say, it starts from a single step. And I think a very outstanding part of your study is you were able to start something, and it's something that has been demonstrated. And a lot of things will follow after that. Just like I was discussing last night when they tried to look at how to be able to detect gravitational waves. When Einstein started his theory, it took 100 years for his theory to be proven. But just imagine, 100 years, and they said that Einstein's theory is not correct, and they will say Einstein is just making these talks. But with all the conglomeration of the technologies that had come, it took 100 years. So, but the most important thing is to take a single step. And I think I am very inspired because I think the implications will not only be for autism spectrum, but for everybody else.
I mean, it's flattering to be, talk about Einstein and talk about us, but I think another really great point from that 100 years of is people trying to disprove that theory did some amazing research and discovered all kinds of like amazing things as well. And so that sort of like that process of like, building off of these studies to try to answer more questions is it's really like, how this progress is gets done.
Yeah, thank you. So I think Jonathan would like to ask a question, but I just like to ask one more because there was slide 12 that I was talking to a while ago, wherein you were making comparisons about the synaptic pruning, the formation of synapses. I had an experience because my advocacy is basically about the gifted children and also about gifted people. So we try to volunteer to help people who cannot be understood by the mainstream, to give them maybe extra education because the mainstream education could not really help them. And plus those who are disadvantaged, we try to help these people. So in one of the volunteer hours that I do, because I also have a background in psychology, I noticed a friend who had her son diagnosed, and yeah, initially one clinical psychologist tried to diagnose her son as having an ODD, of positional defiance disorder, and also on the ASD spectrum. At that time, of course, they said he was having an Asperger's. So I was asking another senior clinical psychologist. Then I said, Doc, I think there is something I could not put on the diagnosis of that doctor, because there's something off. Then that doctor told me, I think the child has childhood schizophrenia, and just watch out when the child becomes an adolescent. So I would just like to put this in question for the information of, many people who might encounter this situation in the future. Yeah, and it's a question in my mind because since that time, my friend did not talk to me anymore. So yeah, I'd like to get your comment about this.
Uh, that is definitely outside of my area of expertise. I apologize. Yeah, I
Yeah,
not.
I get. I can speak a little bit just from the psychiatry realm, I suppose. And so, the way that psychiatry works right now is that there's there's basically a history, there is a mental status exam, there's an interview, and then we come up with kind of the diagnosis that we think are accurate. And so, this is where it gets hard, right? And so, two doctors might have two different opinions, and a third would have another one yet. And so, to me, I'm always very cautious with those things, particularly when somebody is young. And so, there was a little bit of mention of that with the other question about autism. And just to be clear, like, our study and our work just in general that Adam and I and everyone else does, I mean, there's no judgment value put on these things, right? We're scientists trying to help. We're trying to understand the basic fundamental aspects of biology and nature, and then hopefully at some point be able to maybe figure out better treatments and solutions for folks, right? And so, I think that that is key with all these kinds of diagnoses. That being said, I'm very cautious to give diagnoses out. And so, if you said someone has something like autism or schizophrenia, I think there has to be, for me, a high bar to be able to do that because there's a lot of implications behind that. There might be schooling effects, there's social, it's just a lot of things involved in that. So I'm very, again, cautious with everyone, particularly children with those kinds of diagnoses.
Yeah, thank you so much, David. So I think it's getting late on the side of Jonathan, so I'll give him the floor.
Oh, thank you, Cecile. So this is adjacent to your study, and if it's outside of your wheelhouse, please let me know. But in your study, you were looking at synaptic density, which is an anatomical measurement. But you can also have changes in the degree of functional connectivity in neural networks, be they biological or computational. So, first part of the question is whether there are pharmacological pharmacological agents which are known to have a similar functional reduction of synaptic density to the results of your study.
Okay, great question in there. Let me just pull that out for a second. And so, what you said is right, right? So, this is a marker in some ways of anatomy, right? So we're just looking very small, but these are just synapses that are there. And what about the function of it? That was honestly one of the fundamental questions when we started this work before this study in autism, but just in general when the tracer was being developed.
Hmm.
And so there's a paper that came out. Kelly Smart was the first author a couple of years ago, Journal of Cerebral Blood Flow Metabolism, that to answer that question. And so I think they had a really great design. And so what they did is they looked at a checkerboard, right? And so sometimes you see these in MRI studies, and basically what they do is somebody sits in a scanner and then for 5 minutes or X amount of time, there's nothing there, and then they see an image and it has a checkerboard. And it's been understood that in a particular area of the occipital cortex called the V1, that there's a stimulation that occurs there, right? It's been really studied in multiple different imaging markers, and it's pretty consistent. So that's what happened. We put somebody with this tracer, right, a couple people, and ran that checkerboard paradigm to see whether or not there was a change. So it was a anatomy or physiology in that sense, right? Did they change? So it turns out that I went into it a little bit with the models, and there's math, and between that math and the data we get, you can pull some of these things out, and so you can pull out the tracer delivery, which in some ways you can think of as blood flow. We call that V1. And in that paper, they showed yes, the blood flow did change like an fMRI study would. And
Mm.
in fact, fMRI studies do show this, right? They show an increase in activity in that area. We see that with blood flow. But then we looked at the same metric we used here in this study, right? Binding potential. We also looked at another one, volume distribution, and there was no change. And so that's like the proof, supposedly, of how we know that this is not like a marker that's going to change a lot during scan. Because that's a fair question, really. You know what I mean? Like maybe someone is listening to music. I think I saw one of those questions before, or or they're upset or whatever. And is that changing some of this? I don't think so. It's kind of a static marker. And so then the follow up question is, okay, what other things can you use to look at that? And so functional MRI, there's a part of that resting state functional magnetic imaging where yes, you can look at in some ways the amount of blood flow in different parts of the really technically it's the oxygen level of the different parts of the brain at the same time and see do they fall in sync with each other in some ways, right? And you could say that is kind of them talking, or that's the physiology of
Hmm.
that. So could we combine that with this? The short answer is yes, we can do that. You tend to need higher sample sizes for that to work well, but we are working through the data anyway to do that, even in a small sample size, just to give us a glimpse of that. And if one day we're able to get a bigger study out of this and try to prove this the next round, that would probably be part of that study.
Hey, thank you. And then part two of the question, again, dealing with endogenous induction of these kinds of functional analogs of your anatomical synaptic connectivity density. There seem to be credible. This is something that we talked about in a prior room session as well, Cecile. There seems to be credible correlative data indicating that microplastics that go through
I think I lost.
Yeah,
Yep.
I think you dropped off there.
Anyway,
autism
we'll wait.
and Parkinson's.
Jonathan, we lost you
Uh,
for a while. Can you repeat your question?
Sure. How far back do I have to rewind? I'm sorry.
I think I heard microplastics.
Yeah, so there seem to be some credible results, correlative, indicating that quantity of microplastic that has crossed the blood-brain barrier and gotten into the brain, in some samples, up to 0.5% of the weight of the brain tissues, is correlated with higher frequency of, I believe it was Parkinson's, autism, and some other neurophysiological conditions. So, what do you think about other endogenous impacts upon either anatomical or functional synaptic density?
Okay, great. I like that question. I would say it's not endogenous, however, because that would imply that it's actually in the brain. This is exogenous. This is like a toxin that's
Yeah,
coming
coming
in.
in from outside. Right.
Right,
That's what I meant.
right, and so I study different disorders, and I do study Parkinson's disease, and I'll give a shout out to one of my postdocs now, Salika here, who's actually looking through data, and we're getting ready to try to publish a paper on some environmental toxicity, and so it seems like this is not plastics, but it actually is air pollution. And so, I don't want to steal that thunder there, but it seems to be that yes, that when you look at different kinds of environmental toxicity, that I think he's going to be able to show that there's a correlation and a link to dopamine in living people that have Parkinson's disease, which I think is a nice
Mm.
step also. And I think that that is in general one of the, I think when I think of like the future of the brain and the things that I get really kind of excited about for the next 10, 20, 30 years, to me, two big things, right? One of them is environment, right? So we talked about, toxicity, but there's a whole bunch of environmental stuff that's happening. There's global warming, and so all these things have. How does that affect the brain? I think there's more of an interest in it, more of an appetite. So I think that's one of these things. Microplastics and plastics in general, I think pull into that, right? Like Teflon and all those things. And so I think it needs to be studied and understood better. And I would just throw out that my other strong interest that involves directly into autism are the social effects of the brain. And I think just kind of the history of of neuroscience is that we studied brains in some ways as blobs. You know what I mean? There's just this thing, and let's not study what it does. But I like to argue in some of my papers that the brain really is a social organ, and so even though we can do amazing, cool technology like PET and other things, how does that relate when we're discussing it in response to each other, and how does that work? Anyway, so those I think that's a great question. I can't answer microplastics this second, but I think we're slowly making inroads in there. I think that my guess is it's going to be a certain percent of the missing link. For instance, when you look at like Parkinson's disease, only a small amount of the people that have Parkinson's has a genetic component to it that's known, and so in autism, it's very similar. And so there's all this X amount of percent of, well, it doesn't seem to be a clear genetic link in there. What is it? And it seems like environment's going to fill in some part of that.
Thank you very much. That was a great and intriguing answer. I look forward to following this area.
Yeah, thank you, Jonathan.
Thank you.
Thank you, David. So there's a quick question here from LT at the room chat. LT said there are other synaptic vesicle membrane proteins. Any particular reason to use SV2?
Astute question there, LT. And so yes, and so that's exactly correct. There's a bunch of other proteins. In fact, the most famous one is synaptophysin. It's not SV2A at all. And so if you look at like a lot of the preclinical literature or postmortem after folks or someone's passed, they stain those things, and that's the kind of the gold standard. We didn't use that one because how do you get a pet radioligand or tracer up and running? And the truth is that it's really expensive and hard to do. And so the reason why this tracer has kind of a curious name of UCBJ is that there is UCB Pharma. And so they're were our partners in this to develop that tracer in the first for the first time, the first point of that. What was their interest in it? So it turns out that the SV2 A is actually the target for some medications that are on the market, right? So levetiracetam, or Keppra, and brevitiracetam are directly involved in SV2A. And that wasn't known exactly when those drugs came on the market, but a period afterwards. And so that was the reason why we targeted that. Now, to prove that SV2A was at least somewhat as good or as good as synaptophysin, there had to be studies showing that, and there are studies showing that. So I can't say that SV2A is any better, but I think it's at least as good as something like synaptophysin. That's a technical but great question.
Yeah, thank you so much, David. We've been here for 1 hour and 35 minutes, and I would like to ask our speakers how much more time do they have for us in case, yeah, LT, if you have a follow up question.
I It's 9:30 on the East Coast here. I think I have to kind of wrap up. I have little kids, barbarians at the gate, I'm afraid. So I'll probably have to sign off. Maybe one more question if it's there.
Yeah, all right. So,
Same here.
yeah, okay. Yeah, thank you so much, Adam and David. I don't know if there's someone who is asking. No, it's just a comment. So I think I will just ask the last question. Um, what do you think about brain computer
Oh
interface in
boy.
relation to what we study now? and in relation to maybe a possible future treatment for autism. Why am I asking this question? Because most of the brain computer interfaces that are coming out in the market are very invasive. But recently, there is one scientist who came up with a non-invasive one just by using liquid ink. So I'm just very curious.
It's funny. A few people sent me that liquid ink paper. Um, so uh, and that uses EEG, which is kind of the area that I spend a lot of my research time in. And it's the thing. EEG is actually like, so EEG stands is it's an acronym for electroencephalography, which where we can use pretty much the same technology as a guitar amplifier to measure activity from the cortex, like kind of the top layer of your brain, right at your scalp. And people have been using EEG for four kinds of brain computer interfaces now for quite some time. And the reason why, that's not to say that they've been like very, really effective and great. Um, but the key is that they're not invasive, right? Like you don't need to go, you don't need to go inside someone's head to do it. You're putting some wires on their scalp, on their head, with maybe some gel to sort of get the connection good. So I think that what we're finding is that, with really these massive advances in AI, machine learning technologies, that we're getting better at being able to, and this is again, this is like sort of a little afield. I'm not an AI machine learning expert of my expertise. We're figuring out how to use the data that we can get non-invasively more effectively. And that's, I think, going to make, coming down the road in the next couple of years, I think, um, some really, that's where I think we're going to be able to make advances that are going to be applicable for lots and lots of people. Because if in EEG cables, you can get a cap that you can go, put on your head and take off in a minute, right? It's non-invasive. Your head gets wet. That's it. Um, so
Yeah. Uh, Thank you so much, Adam. I think, David, you want to say something?
No, this is Adam's more the specialist here, I think. Just saying, I mean, the one thing I would say is that there was a product a while ago, I think it's called Muse, where people were able to do like, mindfulness or meditation with a EEG like device. And so I think those things have like kind of clear applications to me. I think when we get into the invasive one, this is a different thing. And so, there are like, say, stimulators now for things like Parkinson's disease, and they've made nice progress with those. And I think clearly the technology is going to get more sophisticated, and that's going to be interesting to see. But these are delicate things, and so there's just something that has to be a lot of, I think, care and technology to be able to make sure it's done right.
Thank you, David. Thank you, Adam. So, yeah, I will not keep you up because I know, it's getting late on your side, and we are very privileged to have you here in Quantum Photonics, and we hope to listen to your future papers, your lectures. And yeah, I would like to give you the mic if you have some last messages to the audience.
Just a thank you to you and to everyone out there. I had a lot of fun here. Hopefully Adam did too, and we're happy to come back anytime.
yeah, no, this was great. Thanks for inviting us. Thanks for, yeah, really appreciate it. Thanks for enjoying our work.
Yes, so everyone, let's flash our mics for our guest speakers. So we were giving you an applause. So I don't know if Darcel is here. We'd like to call Darcel up to help us close the room. Are you here, Darcel? Are you able to close the room?
I don't think he can come up. I invited him up a couple of times on the stage, so if I could be Darcel for the moment.
Yes, go ahead, Bob.
All right.
Thank you.
So we're going to close with a nice quick 5 count. And Jonathan, you're going to be number 5. Will you give us a 5, sir?
Cinco.
And Kevin, you're number 4.
4.
I'll be 3 for me. And Cecile, can you give us a 2?
2.
And David and Adam, you are number 1.
One.
All right, 1.
That is
Yeah.
awesome.
Yeah, thank you. You are number 1 indeed, David and Adam. Thank you so much. We hope to see you back in Quantum Photonics soon. Good night.
Thank you.
Good night. Good night, everyone. Thank you.
Thanks,
Happy
everyone. Good
All
night.
holidays.
right. Thanks, bye, everybody. Bye.
You guys
Bye.
rule.